I'm in the last row there? Either of you? Any of you? Okay, great, thanks. Okay. Well, before we get too far into the lecture today, I just want to touch base and be sure that any questions you might still have from nitrogen metabolism. Do you have any questions you want me to address before we move on to the integration of metabolism? Just a reminder that our next hour exam will be Monday in 2 rooms, and I will send a notice about that. And there will be a review session tomorrow at 6? At 6, in here. So those have proven to be useful in the past, so I think I'd encourage you to take advantage of that opportunity if you can. Any other questions either about nitrogen metabolism or about course organization before we launch into the next subject? Okay. So far we've been talking about metabolism principally at the level of a cell at a time. We've talked about regulation and about, at the level of a cell, what might turn things up or turn things down. But once you get beyond being a single cell, then the different parts of the body need to coordinate and be doing the right relevant thing at the right time. Not necessarily the same thing, but the right relevant thing. And so everything above the level of single cells has developed some method of integrating metabolism in ways that will regulate individual metabolic compartments to avoid futile cycles, avoid waste, and to make sure that what's needed in other places is made to happen at the right time. One way in which this happens is through compartmentation. We've seen in cells that the different compartments of the cell have different jobs. The cytoplasm has responsibility for glycolysis and gluconeogenesis, fatty acid synthesis. The mitochondria do a lot of things involved with generating energy. So they do fatty acid breakdown. They do the citric acid cycle, amino acid degradation, some of it. Chloroplasts are specialized for capturing light energy and making sugar. We're going to see some other things as we go into the nucleus. But there's also coordination between organs. And we're going to touch on a few examples of how the metabolism of one organ complements the metabolism of a different organ so that the whole organism benefits from that. And one of the ways in which all of that happens is through using hormones to act as signals for the body as a whole. Hormone as a word means I urge to action, and hormones are molecules that cause action on the part of other cells. And we're going to look a little bit at how they work and how they signal things. We can't possibly go through all the hormones. There are dozens and probably more being discovered every year. But 2 hormones that are of a special interest because of their broad implications and their broad health consequences are insulin, glucagon, and some that are in kind of the same area of regulation. And we're going to look a little bit at diabetes because it's a case history, if you like, of a metabolic disease. It's a disease of sugar metabolism, but also of fat metabolism, and it's one of huge, huge public health impact today. So that's sort of where we're going to be heading. This is an example of compartmentation in the cell, and this is partly regenerated by me, so there's some things going on in here that weren't in there originally. So you can see here in the cytoplasm several aspects of sugar metabolism, glycolysis to break sugars down, gluconeogenesis to make sugars when you need that. Now that, of course, is not in all cytoplasms, principally liver and a little bit kidney. Pentose phosphate pathway is kind of a flexible pathway that can provide reducing power. It can provide ribose. It can provide glycolytic intermediates depending on what's needed. Here's a number of other kinds of synthesis. We see fatty acid synthesis out there. We see nucleotide synthesis out there. Some amino acid synthesis, some amino acid breakdown, and part of the urea cycle. The urea cycle is split between compartments. Inside the mitochondrial matrix, citric acid cycle is this amphipathic cycle, good for breaking down some things, good for making other things. Oxidative phosphorylation, with the exception of one of the, I shouldn't say exception, the oxidative phosphorylation enzymes are principally in the membrane there. Beta oxidation of fatty acids, ketogenesis to turn carbons from fats into water-soluble carriers of energy for elsewhere in the body, various parts of amino acid synthesis and degradation, and the rest of the urea cycle. Now, all those pathways have a lot of components. Some of those components are needed in the other compartment, and so the mitochondrial membrane has a large variety of transporters that permit the movement of the metabolites to coordinate these pathways. Pyruvate obviously would be principally coming in to enter for the citric acid cycle. Carnitine is for carrying the fatty acids into the matrix to be able to oxidize them. Citrate can take it either direction, but you would want it out to make fatty acids, and so on. But beyond just the cooperation of the compartments of the cell, there is cooperation between whole organs. And we're just going to look at a couple of examples of that because there are a lot of them, and we can't cover all of them. But it'll give you a little bit of a feeling for the fact that different organs have different proportions of what they're doing. So to take one example, in anaerobic exercise, when you keep pushing past the point where you can keep up with that with the mitochondria, the demands of energy cannot be met through oxidative phosphorylation. You're going to go through glycolysis, but you can't get rid of the pyruvate through oxidative phosphorylation because the demand for energy is exceeding the rate at which oxidative phosphorylation can run. And in that case, after you've had your glycolytic event, so here's you're breaking down your glycogen to make glucose. Glycolysis is giving you a little bit of ATP, not a huge amount, and making pyruvate. If the effort of the muscle is so great that it exceeds the capacity of oxidative phosphorylation to keep up, then the alternative for the muscle is to convert the pyruvate into lactate. That permits glycolysis to continue because you have to regenerate the NAD+. And then the lactate can leave the muscle through the bloodstream and travel to the liver. And in the liver, in time, at the leisure of the liver, so to speak, the lactate can be converted back into pyruvate and used for gluconeogenesis by the pathways that we're already familiar with. The ATP would be generated in the liver through, for example, beta oxidation of fatty acids, and then that would produce glucose that can go back to the muscle. I think I gave you the example back when we talked about glycolysis, about alligators who put out a spurt of anaerobic activity. They burn up a bunch through glycolysis. They generate a huge lactic acid debt, and they then take half an hour to hours to regenerate the glucose in their livers. And so the net effect of this is energy moving from the liver to the muscle because you're regenerating it by ATP that's invested in the gluconeogenesis to go back to the muscle. Under other circumstances, if you have very sustained aerobic exercise or if you have fasting, muscles may need to dispose of nitrogen. They may be breaking down amino acids in order to provide carbon backbones. Or they may be degrading proteins just out of damage, for example. And so what would be happening then in the muscles? So if you have, under those circumstances, you have a glucose molecule that you've broken down to pyruvate, and the pyruvate becomes the recipient of the nitrogen through transamination. So this is a way to get rid of a nitrogen here that you're trying to dispose of. That generates the alpha keto acid in the muscle. The alanine can leave the muscle, go to the liver, and then the liver will use the alanine as a source of nitrogen for the urea cycle, and the carbon backbone that's left over after that is now pyruvate, and the pyruvate can be used for glucose to go back to the liver. And so the net effect of this is that nitrogen is going to move to the liver from the muscle. Both of these are cycles that involve two organs. One case, you're shifting energy from the liver to the muscle. In the other case, you're shifting nitrogen from the muscle to the liver. In each case, taking advantage of complementary metabolic pathways in the two locations. You notice here how the glycolysis and the gluconeogenesis are happening in different organs, and that was true in the other case as well. But there are really a large constellation of metabolic events that need to be coordinated. And in doing this, the body makes use of a group of molecules called hormones. And any molecule that can act from one place to another place can be considered a hormone. There are some hormones that are generated in the brain and circulate through the whole body. There are others that act more locally. But if it's a molecule moving from one cell to tell another one to do something else, you can consider it a hormone. But of course, molecules by themselves don't do very much. And so there has to be some way for the chemical structure to inform the activity of the cell that it's talking to. And we call that signal transduction. Signal transduction is the name for the series of processes by which a chemical can be turned into action at the bottom of the chain. And signal transduction has some common features pretty much for whatever hormone we're talking about, with some modifications, but some general features. There's always some kind of a ligand. So ligand just means the thing that binds. This is our generic name for a hormone or for anything else that binds to a receptor. There needs to be a receptor, and that receptor has to have some kind of a ligand binding domain. Many hormones use transmembrane proteins. There are a few hormones that don't, but many hormones use transmembrane proteins. And so the ligand binding domain will usually be outside the cell, and there'll be some kind of an intracellular domain talking to the cytoplasmic face. There will be, generally speaking, some kind of a change in conformation when the ligand binds. That will lead to changes in some intracellular components. Quite frequently, there will be the generation of some kind of a second messenger that will amplify things. Depending on the number of these intracellular components, you may get a cascade with amplification at every step. There'll be targets either out in the cytoplasm or in the nucleus. Somehow or other, you're either going to change metabolic activity or change gene expression. You need to change something for it to have an effect. It could be something short-term, like metabolic activity, or something longer-term, like gene expression. And there's always an off switch. I don't think your textbook mentioned the off switch, but there are always off switches. In biology, you don't do anything forever. Let's see, Doug's pretty good. So let's think a little bit about protein kinases. These are a big team in the hormone story. Protein kinases are kinases. They transfer a phosphate onto a protein. So protein kinases are kinases that target some other protein, sometimes themselves, but they target proteins. And there are two major families of protein kinases. Some of them add their phosphate to a tyrosine. So here you can see a phosphotyrosine, its structure. Some of them target serine or threonine. Typically, that kinase will target either a serine or a threonine, but there's some structural similarities between those families. And it's worthwhile noting that in both of these families, whether the tyrosine or the serine-threonine ones, they're both of them changing a neutral side chain to a charged side chain. So you're going from something that's small and hydrophilic, but just polar, to something that's fairly bulky and charged. Has two negative charges on it now. So the analogy I think of for protein kinases is you just added a phosphate, but it's like putting a doorstop on a door. The doorstop may be 2 inches long, but it can hold the whole door open because it's in the right place and resistant. So insulin is one of the premier hormones. It was the first one that was well understood. It's one that has huge public health impacts, and it allows the body to regulate which metabolic fuels it's going to burn as a function of fed state or of fed versus fasting. And this is showing you some of the variations in which fuels are being used at which time. So shortly after a meal, a typical person will be getting half their energy by burning carbohydrate. a third from burning fatty acids, and maybe about a seventh or so from burning proteins. After an overnight fast, so after a 12 to 14-hour fast, you'd have the proportion of energy coming from carbohydrate goes way down. The proportion coming from fatty acids has more than doubled. And the proportion coming from amino acids is just starting to change. This is a relatively short-term change. It will change as soon as people eat breakfast. If you continue to fast, for whatever reason, there's continued metabolic adaptation. And by 40 days of fast, essentially zero. There may be trace amounts of carbohydrate at that point, but a very large fraction coming from fatty acids, typically in the form of ketone bodies, and now a little bit of amino acids. Your body is going to try to protect those amino acids to the extent that it can. But you can see if you keep burning protein for 40 days, long-term consequences come about. Do you have a question there? Okay. All right. Oh, sorry. Well, how long can you survive with water and no food? 40 days is listed as sort of towards the outer limit. It's going to depend a lot on your condition beforehand and what you do during it. During World War II, a number of volunteers who were conscientious objectors were volunteered for studies of starvation and refeeding. The American government knew there were going to be people who had suffered severe deprivation and wanted to know how to safely refeed them. And these people were put on like 500-calorie-a-day diets and lost like close to a third of their body mass or a quarter of their body mass. And so the studies were done on metabolic changes and how well and quickly they could refeed and so forth. Typically, people who are fasting will reduce their activity. They will do everything they can to put on more clothes to stop losing heat, because that's a source of energy. People become compulsive. Their thoughts become completely occupied with thinking about food. And what can kill you in a fast is actually your heart, because the body will use muscles. It'll use skeletal muscles first, and then it'll start using heart muscle. So it can be a very, very catastrophic thing to keep up a fast for too long. It's possible to have a modified fast of, say, 500 calories a day for a very long time if it's under close medical supervision, if it's very carefully monitored, if very careful composition of the diet is taken, so that you can have the body draw down the fat stores without taking the muscle. But it's not something to be undertaken just casually or without really close supervision of someone who knows what they're doing. So insulin is one of the hormones that helps the body keep all that juggling straight. And the things that it does at the metabolic level make sense in terms of what it's trying to accomplish. So if you think about the usual circumstances under which insulin is released, insulin will rise as the blood sugar levels rise. Typically, when you eat a meal, your blood sugar levels will rise. Insulin will be released in response to that. And it's a signal that the body is fed, that energy is available. And so insulin will act on many cells to accomplish many things, but all of those things make sense in terms of thinking of insulin as the marker for the fed state. So in muscle cells and many cells, insulin will increase glucose transport. It will increase glycogen synthesis. I've got some spare sugar. Let's put it in the bank in the form of glycogen. And complementarily, it will suppress glycogen breakdown, so you don't get a futile cycle. Adipocytes, of course, are the fat cells. They're the ones that are specialized for fat storage. So insulin will target them, but for other things, extracellular lipoprotein lipase is the enzyme that allows the cell to get fatty acids off of LDLs. And it will stimulate acetyl-CoA carboxylase, which is that rate-limiting step in fatty acid synthesis, so that the adipocyte is geared up for making fat out of sugar and out of protein. And it will suppress fatty acid release. The fat cell is supposed to let go of its fatty acids when things are needed, but insulin is a sign that things are lush and rich, so don't let go of it now. Insulin is made in the pancreas. The pancreas is a small organ near your liver that is involved in general with making things for digestion, so it makes digestive enzymes. But it has little islands of tissue scattered through it that stain differently. You can see this little pink island here. This is a beta islet, and these are little islands of endocrine tissue embedded in the tissue that makes the digestive enzymes. And so the beta cells make insulin. And interestingly, inside the pancreas, it is the metabolism of glucose that constitutes the intracellular signal about glucose levels. So you wonder, how does the pancreas know how much insulin to release? Well, it's actually done by exploiting, interestingly, enzyme kinetics. So there was a reason you learned them all that while back. And this is an example of the relative activities. So when we look at the activity of hexokinase, which is the first step in glycolysis in all cells, has a fairly broad spectrum of which hexoses it can phosphorylate. Hexokinase has a very high affinity for glucose, and hexokinase is operating at pretty much its Vmax at any blood glucose level in which you'll still be alive. But there's another enzyme called glucokinase. It catalyzes the same reaction, but it does so only for glucose, and it has very different kinetics. And what you can see here is that because of its lower affinity, it has an almost linear response as the blood sugar is rising. So the glucokinase acts as a glucose sensor in order to detect changes in level in the blood sugar. This is also true in the liver itself, which has glucokinase. Interestingly, glucokinase is one, if you notice this isn't quite a straight line here, it's got a little bit of a sigmoid character to it. Glucokinase is one of those rare things of a monomeric protein that has sigmoidal kinetics. And they think that that's because it takes a while to relax back into its previous state. That's called hysteresis. That it having catalyzed the reaction once, it keeps some affinity and before it relaxes back into a lower affinity state. So it's an interesting example of enzyme kinetics actually having a significant impact on day-to-day health. They do elsewhere, but this is a striking example of it. So that's the glucose sensor inside the pancreas, and it will release insulin out into the environment. And out in the environment, it passes through the blood to encounter lots of cells. In order for insulin to affect a cell, the cell has to have insulin receptors. If no receptor, the cell is blind. You could have a ton of insulin out there. And the insulin receptor is a dimer of dimers, an alpha-beta dimer. So there's 4 peptide chains altogether. The alpha chain is a large extracellular domain, and then the beta peptide is the transmembrane that allows there to be communication from the inside to the outside. When insulin is bound, that then changes the conformation of the peptides, and the beta chains can interact with each other to do what's called autophosphorylation. It's a little misleading. It's not that one peptide chain phosphorylates itself, but that one beta chain of the receptor phosphorylates the other beta chain, and then that one may return the favor like that. So the receptor phosphorylates itself. One peptide phosphorylates the neighbor peptide. This is a tyrosine kinase activity, and it's intrinsic to the beta chains of the receptor. There's another component that's also required for this to happen. This general behavior of having a transmembrane protein and a tyrosine kinase is a very common strategy for peptide hormones. There are a large number of growth factors that are actually short peptides or medium-length peptides. And many of those peptide growth factors act through tyrosine kinase receptors. Yeah. Any questions so far? Okay. So let's take a little look at some of those effects of insulin. I've mentioned that there has to be a receptor for it to bind. In both muscle and adipose cells, it will increase glucose transport. And they do it by kind of a cute trick. Let's see. It doesn't look too bad there. Okay, not perfect, but there are insulin receptors inside the cell that are already structurally correct, but they're in vesicles that are a closed, dead-ended compartment. They're ready to go, but they're not in contact with the cell membrane. And the consequence of exposure to insulin is that these little vesicles can merge with the plasma membrane, with the cell membrane, and now the transporters are exposed to the blood and can let more sugar through. So if you have, say, 5 receptors before you're exposed to insulin and you have 15 after you're exposed to insulin, 3 times as much glucose can enter the cell. So you can stimulate glucose transport by just changing the number of transporters. It can increase fatty acid uptake by increasing the lipase activity so that it's cutting off more fatty acids from the LDLs that go by. We mentioned that insulin can modulate glycogen metabolism, that it needs to turn up the synthesis and turn down the degradation. It is true that there is some allosteric regulation of the glycogen enzymes, but a significant part of it happens through kinases. And so you have the 2 enzymes of interest, the glycogen synthase to make glycogen, and glycogen phosphorylase, which will split glycogen with a phosphate to degrade it. And so the kind of reciprocal tuning that goes on looks like this, that you can, for example, take an inactive glycogen synthase and dephosphorylate it to make it more active. And you can take the glycogen phosphorylase, dephosphorylate it, and make it less active. So taking off the phosphate has a different effect depending on the enzyme you're doing it to. After you've been, several hours after you've eaten, maybe after you've fasted overnight, you want to change that balance. Different protein kinases can add a phosphate to the synthase and turn it down. And they can add a phosphate to the phosphorylase and turn it up. So the regulation up and down is by protein kinases, but different kinases under different circumstances. But it's always a reciprocal regulation. So that's the hormone that dominates our metabolism for several hours after we eat in the fed state, the replete state. We're not all eating all the time. And after enough hours have passed and the blood sugar is going down, it might start to be appropriate in the body to recruit some of these fuels and to start getting them out of their stores. So the complementary hormone to insulin is glucagon. And glucagon is also made in the pancreas, and its name means I urge sugar. So it's the hormone that can bring sugar out of its stores and into other things. So the liver can mobilize its own glucose stores. It has some sugar stored as glycogen. It can recruit those. Some of the other hormones that may be involved in recruiting fuels are epinephrine and norepinephrine. Glucagon's targets in the liver would include glycogenolysis, so turning on the glycogen phosphorylase. It might include gluconeogenesis. So in the liver, it would be using the carbon backbones from amino acids to make new sugars. Go away. Don't want you. There we go. Visible. There we go. In adipocytes, it would be stimulating the hormones that break down the triacylglycerides. Epinephrine and norepinephrine are more targeted to other tissues. So, for example, they will stimulate glycogenolysis in the muscles to help recruit the glucose that's stored in glycogen. The structure of the glycogen receptor is somewhat different from that of the insulin receptor. If you look at it, it's structural in a ribbon diagram. This is embedded in a membrane now. So imagine that the membrane runs here and here. And if you count these, you'll see that there are 7 of these transmembrane helices. And this is called the 7-TM receptor family, for the 7 transmembrane domain family. This is another very common strategy, just like the tyrosine kinase receptors were. Some of the examples of other members of the family include the adrenergic receptors, rhodopsin, which is the light-sensing protein in the membranes. And when it binds the ligand, when it binds glucagon or whatever else, it again changes configuration, but this is not a tyrosine kinase the way the insulin receptor is. Instead, there are other transducers that are going to get the signal across. Many receptors interact with a group of sort of transducers that are called G proteins. They're called G proteins because these are small proteins that can bind GTP or GDP. And the cycle of activation looks something like this. If the G protein has a GTP bound, it's in the active state. It can activate something else. It will continue to do that until it hydrolyzes the GTP and sheds the inorganic phosphate, and then it's an alpha GDP, and it will pick up a beta gamma subunit that will combine with it and inactivate the alpha subunit. They'll remain inactive until something stimulates it to exchange the GDP for GTP, and then the complex will break apart again into two parts that can go and do things. This process of hydrolyzing the GTP or exchanging GDP for GTP can both of them be accelerated by accessory factors that are called things like GEFs and GAPs. G would be for GTP exchange factor. GAP would be the it's the one that accelerates the hydrolysis of the phosphate. I don't remember what the acronym stands for. I apologize. All right. So the alpha GTP, the G protein with the GTP bound by it, can then interact with another protein, adenylyl cyclase. And adenylyl cyclase is another transducer molecule, if you like, that comes in an active and an inactive state. In its inactive state, it's a tetramer with regulatory, R for regulatory and C for catalytic subunits. If it activates the adenylyl cyclase, it can then, I'm sorry, this is the kinase we're talking about here, excuse me. Adenylyl cyclase is the enzyme that can take ATP and make a cyclic AMP out of it. So the alpha GTP is activating the adenylyl cyclase. And what's interesting here is you've taken an ordinary ATP molecule, throwing away a pyrophosphate, and you've got this cyclic form down here in the corner. And you can see that that looks different. And it's sufficiently unstable that it makes a good temporary marker because it won't hang around forever. This molecule, the cyclic AMP, it's an AMP, an adenosine monophosphate, but it's cyclic because of here. This molecule, the cyclic AMP, is the one that's going to target the protein kinase A. And protein kinase A is the tetramer of two catalytic units and two regulatory units. When cyclic AMP interacts with the regulatory units, it liberates the catalytic units, and then the catalytic units are capable of not attacking, but targeting serines or threonines, depending on which particular target they want to be in that cell. So, for example, phosphorylase kinase is the enzyme that's going to attack or modify phosphorylase, and the phosphorylase kinase is one of the substrates for the active protein kinase A. It's called protein kinase A because it's cyclic AMP that activates the regulatory subunit. So that's where that name comes from. Now, I mentioned things have off switches. We're not going to go through the off switches for everything, but this is one of the off switches. Here's your cyclic AMP. Phosphodiesterase can split this bond. That's actually a phosphodiester. And now you get back AMP. Interesting fact, caffeine inhibits phosphodiesterase. One of the reasons that caffeine has some of the effects it has is that it's making the cyclic AMP hang around longer. Protein kinase A in a different cell in the fat cells has different targets. There are hormone-sensitive lipases, and it can move those lipases onto the fat droplets so that it can start to break down the triacylglycerides to get more energy out into the bloodstream. So, and this is a comparison for you of two different G protein coupled receptors, showing you how parallel a lot of the structure is and how it depends on the intracellular targets, what the outcome is going to be in that particular case. So in both cases, we're starting with either glucagon or epinephrine. So these are both the hormones that can mobilize energy. Both of them have act through a G protein coupled receptor. When the ligand binds that, it activates the G protein. The G protein then in its turn can activate the adenyl cyclase. The adenyl cyclase can generate cyclic AMP. The cyclic AMP can then target protein kinase A. Now you notice that this is a hormone, this is an enzyme. And so one molecule of G protein, by activating an adenyl cyclase, can make dozens or hundreds of molecules of cyclic AMP. This is one of the amplification steps in the process. This cyclic AMP activates the protein kinase A, and each protein kinase A can activate dozens or hundreds of target proteins. But look at how it differs now in the two cell types. If you were looking in a muscle cell, for example, the protein kinase A would have as its targets phosphorylase kinase. Phosphorylase kinase could then phosphorylate glycogen phosphorylase, and that's going to then break down glycogen, liberating glucose. And it would also phosphorylate glycogen synthase. But the phosphorylation here turns it off. So the protein kinase A is acting the same way on two different proteins, but having opposite effects. Whereas over here in the fat cells, the protein kinase A has different targets available to them. There's essentially no glycogen in a fat cell, but there are lipases, and now it can lead to lipolysis. So a lot of the machinery is shared. There's the possibility of amplification in both cases, but the outcome will depend on the cellular targets. Any questions so far? G proteins occur in a lot of other signaling pathways. This is a common strategy for signaling about one or another event. And to give you a feeling for just how common it is, 1% of the genes in the human genome are G-linked receptors. So this is a very broad and flexible strategy to be used. G proteins can do more than just activate adenyl cyclase. Depending on the cell and depending on which G protein, they can inhibit cyclic AMP. There's a parallel enzyme called guanylyl cyclase, which can make cyclic GMP, right? And there are other kinases and lipases as well that may be mediated through a G protein. This is an example of a cyclic AMP-gated ion channel. So G proteins can be involved in ion flux and therefore in neural transmission and so forth. The G proteins also play a part in transduction from epinephrine, but through a somewhat different downstream pattern. And that's the phosphoinositides. This is kind of a cute trick. This should look at least a little familiar to you. This is phosphatidylinositol, bisphosphate. So we know about phosphatidylinositol. That's the fatty acyl tails over here, and this is the inositol phosphate. But this is also called PIPP because it's phosphoinositide with 2 extra phosphates. And in part of this signaling cascade, an enzyme called phospholipase C, so this is a phospholipid, and it's phospholipase, is going to split this molecule into the portion that's just the acyl part, and then another part that's the inositol triphosphate. You can see this is inositol in here and 3 phosphates there. And so phospholipase C is taking this molecule that is, potentially just part of the membrane or something like that, and splitting into something that's very hydrophobic and very hydrophilic. It's sort of like letting them split apart. And we're going to see that both of those compounds turn out to be important in signal transduction. So we talked already about the glucagon and epinephrine pathway that went through the G proteins through adenyl cyclase and ultimately to a lipase. Epinephrine has open to it additional kinds of receptors on different cell types called the alpha adrenergic receptors. These also use G proteins, but in this case, the G protein is activating that enzyme I just talked about, phospholipase C. Phospholipase C gets its name because the C stands for calcium. It's a calcium-activated phospholipase. So when phospholipase C splits the phosphatidylinositol, we have this highly charged inositol triphosphate and this very lipid-soluble diacylglycerol. And each of them have effects on things. The diacylglycerol can affect protein kinase C. The IP3 can affect calcium channels to liberate calcium. And that calcium can signal to a couple of different things themselves. The calcium will further activate phospholipase C, and it'll activate protein kinase C, C for calcium. And then that will have, excuse me, whatever happened there? Okay, there we go. Other cellular responses. So again, we can see the downstream effects are going to depend on the downstream cascades. And I just want to point out that there are all these kinases going on and so forth. Phosphatases also matter. We're not going to discuss them in the same detail, but obviously everything that got a phosphate added probably at some point is going to have the phosphate taken off, and that's done by phosphatases. So that brings us to diabetes. And we're going to talk a bit about diabetes as one example of a metabolic disease. And it's probably a good general way of thinking about diabetes is as a disease of energy metabolism rather than as a disease of glucose per se. Diabetes presents or catches people's attention as a couple of things. One is of there being sugar in the urine, and the other is of excessive urination, that diabetes means flowing through. And even the Greeks noticed that someone with diabetes needs to urinate a very great deal. What the body's trying to do is to dilute the sugar and get rid of it any way they can. We can distinguish between two kinds of diabetes. I should say that this is diabetes mellitus, the sugar-associated kind of diabetes. There's a separate kind of rarer diabetes called diabetes insipidus, where you also urinate a lot, but that's a difficulty with the kidney. But the kind of diabetes you're almost certainly going to have dealt with is diabetes mellitus. Let me just stop and ask, how many of you, wait, let me say the whole question before you raise your hand. How many of you either have diabetes, have a family member with diabetes, or have a friend with diabetes? Can you raise your hands? Take a look around. This is a major, major health problem. My great-grandfather lost a leg to diabetes. My mother had it, so I have family connections of my own. There are two kinds of diabetes that present. One form is insulin-dependent, meaning the person doesn't make enough insulin, but if you give them insulin, they're okay. And this is an autoimmune disease. It turns out that if you can catch the earliest symptoms and treat them with immune suppressants, in many cases, you can actually arrest the disease and give the body a chance to recover from the autoimmune insult. We used to call this juvenile diabetes because type 1 diabetes typically presents in people under the age of 25 and can be in their teens or younger. The second type of diabetes is called type 2. This is insulin independent, and it's called that because these individuals have insulin in their blood, but their cells aren't paying attention. So whether or not you change their insulin levels, that won't be a very effective way of changing their blood glucose. We used to call that one adult diabetes, and it results from insulin resistance. It's someone who has developed over time a metabolic alteration in their cells such that they no longer listen to the signals of the insulin that's being presented. I say used to be called juvenile and used to be called adult. When diabetes was first being understood at the beginning of the 20th century, the type 1 insulin-dependent form was almost the only kind of diabetes that was seen in anybody under 25. And type 2 diabetes only turned up in adults. Type 2 diabetes is strongly associated, not exclusively, but strongly associated with being overweight. And there simply weren't enough severely overweight children in 1920 for there to be a significant number of type 2 diabetics among children. That, unfortunately, is changing. The figure legend on this figure in your textbook was a little ambiguous. It is the dog that has the diabetes and not the boy. This is a picture of Banting and Best. Banting and Best were Canadian physiologists who were the ones that were able to identify the pancreas as the organ that makes the hormone that regulates blood sugar. And this is the dog that was the first experimental subject in which they were able to induce diabetes. Okay. Trying to decide how far to get into this today and how much to leave for Friday. I'll cover a couple more slides maybe, and then we'll call it quits. Diabetes has its effects on the body because the cells are not taking up glucose. We mentioned that the glucose transporters are modulated by insulin. If you don't have enough extra glucose transporters, the blood sugar stays high, and you have hyperglycemia. You have high upper sugaremia in the blood. You have high blood sugar. And because the sugar's out in the blood and not getting into the cells, the body actually ends up thinking that it's starving. And so, for example, it may attempt to do more gluconeogenesis. So the liver is actually adding more sugar to the bloodstream at a time when actually there's plenty of sugar out there. The adipocytes think that there's not much energy around, so they try to hydrolyze their lipids, and you have elevated serum lipids. The levels of sugar can get so high in someone with severe diabetes that there can be non-enzymatic modification of the proteins. That is, the proteins in the blood can have glucose molecules added to them simply from the sheer volume of glucose out there. No enzymes are involved. It's just a reaction. All right. Okay. All right. I guess we're wrapping up. I'll see you Friday.